So, the central dogma of molecular biology. Now, for our learning outcomes, first is to summarize the requisite molecules for initiation of replication, transcription, and elongation. Next is to elaborate on the mechanisms and step-by-step reactions of the processes in the central dogma of molecular biology. Next, point out the differences between prokaryotic and eukaryotic transcription and translation, and illustrate the resulting RNA transcript and amino acid sequence of a polypeptide based on a given DNA template following the principles in transcription and translation. So, for the outline of discussion, we will discuss the flow of genetic information, replication, reverse transcription, and finally translation. So, for the flow of genetic information, Sir Francis Crick's statement in 1970 outlines the central dogma of molecular biology, emphasizing the precise transfer of sequential information at the molecular level. According to this principle, information flows from nucleic acids to proteins but not vice versa. He specifically said that the central dogma of molecular biology deals with the detailed residue-by-residue transfer of sequential information. It states that such information cannot be transferred from protein to either protein or nucleic acid. Okay, Francis Crick, co-discoverer of the structure of the DNA double helix along with James Watson, titled the sequence of events from DNA to proteins as the central dogma, keeping in mind that the meaning of dogma is a point of view that is presented authoritatively without the need for any proof. Okay. The central dogma of molecular biology specifies the processes central in a typical cell's natural life, growth, and development. This involves the DNA macromolecule that contains the genetic information and functions to control directly the synthesis of proteins. This refers to Crick's sequence hypothesis, which states that when the information has passed into proteins, it cannot go back to either DNA or RNA. Furthermore, Crick emphasized that the transfer of information to and from nucleic acids and from nucleic acids to proteins is possible, but not from proteins to proteins nor proteins back to nucleic acids. This means once information is encoded into proteins, it cannot revert back to DNA or RNA. Crick also highlighted that while information can transfer between nucleic acids and proteins, it cannot occur between proteins or from proteins back to nucleic acid. So, figure 1.1, this is the central dogma of molecular biology. DNA replication, ito class, from DNA to DNA, ito yung process niya, replication. And then DNA to RNA, we call that transcription. And then from RNA to protein, that is what we call translation. The genetic information that the DNA contains lies in the sequence of bases and that specify a gene. Now, what is a gene class? A gene is a physically defined segment of the DNA. That specific segment in the DNA containing the unique sequence of nucleotides corresponds to either 1. The equally unique sequence of amino acids in a polypeptide or protein, or the nucleotide sequences in an RNA. Next slide. Since the information is inheritable or can be transferred to the offspring, it is well suited to be called genetic information. The DNA, therefore, bears the function as a repository of genetic information. This is preserved and inherited by the next generation by a faithful replication mechanism involving many enzymes. One replication event results in 2 double-stranded DNA. DNA does not only store the genetic information, but is also used for gene expression. Gene expression starts when RNA molecules are synthesized in a process called transcription. In the transcription process, messenger RNA or mRNA molecules that are produced serve as templates for protein synthesis. The mRNA sequence that resulted from copying the DNA gene segment during transcription is now the code that will help synthesize and put together the correct amino acid in a protein or polypeptide. The synthesis of a polypeptide or protein is called translation. Now, the figure shows the processes of replication, DNA to DNA, transcription, DNA to RNA, and translation, RNA to proteins. Following the discovery of retroviruses, or viruses that contain RNA as genetic material, and the enzyme reverse transcriptase, also known as RNA-dependent DNA polymerase, recent representations of the central dogma contain the reverse arrow, from RNA to DNA. You call that process reverse transcription. Okay? DNA replication is a process that synthesizes DNA by copying existing DNA. This phenomenon happens before cell division and ensures that daughter cells will result with the same structure and amount of genetic material. The DNA in the cell remains essentially unaltered through the lifetime of the cell and in subsequent generations. So class, before cell division, synthesis phase, right? So dyan nangyayari ang process of replication. Although occasionally, the cell experiences shuffling of information as regions of the DNA are exchanged between chromosomes during cell division by a process known as recombination. This creates genetic variation that brings about evolutionary changes in molecules, proteins in particular, and in organisms. Okay, what is semi-conservative DNA replication? Replication in all species follows the semi-conservative model, as evidenced in the Meselson and Stiles experiment. In this mode of DNA replication, the original or parental strands separate, and each becomes a template for the synthesis of a new strand. The two progeny molecules, therefore, contain the conserved original or parental strand hybridized to the new or daughter strand. So what are the requirements of the replication process? The first requirement for the replication process is the double-stranded DNA or dsDNA. It begins at a fixed point in prokaryotes, and ori is the designated point of origin in their circular chromosome, where a replication bubble is first observed. Class, shown here, ito yung ori or origin of replication sa prokaryotes. Tapos this is the replication bubble. Okay? For higher organisms such as in eukaryotes, multiple replication bubbles are seen as shown here. This is our first replication bubble. Ito yung pangalawa. Ano? And the synthesis of the new DNA occurs at the replication fork created by those two bubbles. Saan yung replication fork ng eukaryotes? Dito class. Dito ang replication fork. Here and there. Tapos for prokaryotes naman, dito yung replication fork niya. Okay? So, the replication fork advances as the unwinding of the parental strands happen. Prokaryotic and eukaryotic DNA with advancing replication forks. In prokaryotes, only one replication bubble is observed as against eukaryotes with multiple replication origins. Ito class, ito yung prokaryotes, isa lang yung replication bubble. Saan siya nag-start? Sa ori or origin of replication. As you can see here, replication fork dito and then here. Versus sa eukaryotes, there are multiple replication bubbles here, there. Tapos same, meron din siyang replication fork. Okay? So, the phosphodiester bond. As a polymer, DNA is made up of nucleotides that are linked together by a 3' and 5' phosphodiester bond. So, class, asan yung phosphodiester bonds? Ito, class. Ito yung side rails ng DNA. Yan ang tinatawag na phosphodiester bond. Now, the nucleotides used in the replication process are in their triphosphate forms: ATP, GTP, CTP, and TTP. They are added to the growing DNA chain by a nucleophilic attack of the electrons in the oxygen of the OH group of the 3' carbon of the growing chain of nucleotides. Dito, class. Nucleophilic attack sa third carbon, dito. To the phosphate of the 5' carbon. 1, 2, 3, 4, 5. Dito, class. Yung 5' carbon ng incoming nucleotide. Now, the release of pyrophosphate provides the energy for the reaction to proceed to completion. So, multiple proteins are required for the replication process. For prokaryotic replication, these are, very important, yung helicase. Helicase is an enzyme that unwinds DNA strands ahead of DNA polymerase. Next, topoisomerase is an enzyme that relieves the torsional stress that arises ahead of the replication fork when the helicase enzyme unwinds the DNA strands. Primase, it is an enzyme that copies a DNA template strand by making an RNA strand complementary to it. The RNA serves as a priming site where DNA polymerase can begin to synthesize a DNA strand. DNA polymerase 3, it is the major polymerizing enzyme and catalyzes the formation of the phosphodiester bond. It can only continue synthesis and lengthen the DNA strand in the presence of the RNA primer. Now, what is SSB? This is a single-strand DNA binding protein. This binds single-stranded DNA to stabilize it so that the hydrogen bonding surfaces of the DNA bases are spatially oriented toward the incoming nucleotides. Next enzyme, sliding clamp. A sliding clamp is a protein dimer that encircles the DNA strand and helps hold the DNA polymerase to the DNA strand. Clamps and clamp loaders, protein B subunit from the DNA polymerase 1 holoenzyme complex, hold the polymerase at the DNA. The sliding clamp helps the DNA polymerase complex to stay on the DNA through an entire cycle of replication. A multi-subunit entity called the Y complex or gamma complex functions as the clamp loader. That is, it loads the clamp onto the DNA. In eukaryotic cells, a multi-subunit protein called replication factor C or RFC is the clamp loader, and the proliferating cell nuclear antigen or pCNA is the sliding clamp. Next enzyme, DNA polymerase 1. This is the enzyme that has an exonuclease function to remove the RNA primers and the polymerase function to replace the primers with the deoxynucleotides. Next enzyme, DNA ligase. This catalyzes the covalent joining of the individual pieces of the lagging strand. It covalently closes nicks in double-stranded DNA. The nick must contain 3' hydroxyl and 5' phosphoryl termini. And the nucleotides being linked must be adjacent in a duplex structure and properly base paired. DNA ligase functions to seal Okazaki fragments in the lagging strand of DNA replication. So, what are the steps in the replication process? Let's discuss each. Let's begin with initiation. DNA replication starts at specialized sites called origins of replication. Yung kaninang ORI class na inano natin diniscuss, and moves away from the origin in both direction. So bidirectional siya class ang movement niya, creating a structure known as replication bubble. Note class, ito yung replication bubble. Adjacent to it, yung replication forks natin, here, tapos dito. Okay? The DNA double helix is opened at the origins of replications and unwound on both sides of the origin to form two structures called replication forks that unwind the double helix in opposite directions. The replication forks are the sites at which single-stranded DNA is exposed and at which DNA synthesis occurs. Now, once the DNA double helix is opened, replication enzymes and proteins are loaded to the single strand, and these will form the templates for the daughter strands that are to be synthesized. After the replication machinery is in place and the DNA has been opened up, replication enters the elongation phase. So next phase na tayo. But before we proceed with the elongation phase, may papakita kong video class in a while.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Right. So, start na tayo. Elongation tayo, class. During the elongation phase, the replication machinery moves along the parent DNA strands and forms the daughter strands as it proceeds. So, termination of DNA replication occurs when the two replication forks moving in opposite directions meet and the replication complexes are disassembled. In bacteria, this occurs at specific site called or termination, whereas in eukaryotes, no specific termination sequence is required. Class, the replication process is kept as single-stranded DNA binding proteins, allowing the polymerases to read the template and put the nucleotide to the growing chain based on a correct base pairing with the template. Base G always pairs with C, and A always pairs with T, Chargaff's rule. The pairing is facilitated by hydrogen bonding or H-bond between base pairs. 3 hydrogen bonds for GC pairs and 2 for AT pairs. The formation of the new DNA chain is always 5' to 3'. The two strands of DNA double helix are complementary and anti-parallel. One is 5' to 3' and the other is 3' to 5'. During the replication event, the synthesis of a new strand on the two DNA template strands occurs simultaneously. However, one strand is synthesized continuously, and this is labeled as the leading strand. On the other strand, known as the lagging strand, is synthesized discontinuously, where discrete Okazaki fragments are formed. An Okazaki fragment consists of covalently linked RNA and DNA fragments. Once the templates are exposed, short, removable RNA primers are synthesized first. They must also be complementary to the template. Hence, whenever there is base A, the complementary nucleotide with base U is attached. Uracil. DNA polymerases cannot initiate synthesis, but can only continue with the free 3' OH in the last nucleotide of the growing chain. DNA polymerase 3 in prokaryotes and the DNA polymerase D and E in eukaryotes continue on with the synthesis of the new DNA strands for both leading and lagging strands. The short RNA primers are removed and replaced by DNA. The nick, or single-strand breaks created by the replacement of the RNA, are sealed by DNA ligase. So class, ito illustration lang to ng replication process. Pero mas better na exemplified dun sa video. Basahin nyo lang to class, please. Okay, DNA replication pa rin to. Okay. What is reverse transcription? Retroviruses are small RNA viruses containing RNA as their genetic material. HIV, the virus that causes acquired immunodeficiency syndrome or AIDS, is an example of a retrovirus. A simple view of the retrovirus life cycle is shown in Figure 9. An essential part of the retrovirus life cycle is converting the RNA genomic material back to DNA. This involves an RNA-dependent DNA polymerase called reverse transcriptase, which uses RNA as a template to make DNA. Like other RNA-dependent polymerases, reverse transcriptase is error-prone because it contains no proofreading activity. This enzyme is part of the viral components that are transferred to the host cell. The replication scheme for HIV is as follows. First step, the viral RNA in the host cell base pairs with a specific tRNA molecule to provide a primer for DNA replication. Next, Reverse transcriptase makes DNA toward the 5' end of the viral RNA. Third step, RNase H partially degrades RNA from the RNA-DNA duplex, removing the 5' terminus. Next, the 3' terminus of the viral RNA base pairs with the overhanging DNA strand, forming a circle-like structure. Next step, reverse transcriptase makes a DNA copy of the entire genome using the 3' end of the DNA strand as a primer. Sixth step, the RNA strand of the duplex is removed and a DNA strand complementary to the first DNA strand is synthesized to form a dsDNA or a double-stranded DNA. And then last step, the double-stranded DNA integrates into the host chromosome with the help of a viral enzyme named integrase. Transcription. Next step na tayo. Transcription is the synthesis of RNA molecules using a double-stranded DNA or a dsDNA as the template. Similar to the replication of DNA, nucleoside triphosphate substrates are connected to one another by a 3' to 5' phosphodiester bond synthesized by a DNA-dependent RNA polymerase from the 5' to 3' direction. Two major differences between transcription and replication are as follows. With few known exceptions, only one DNA template strand is transcribed. This is the template strand, also known as the non-coding or antisense strand. Pangalawa, only a small fraction of the entire genetic material of an organism is converted to RNA in one cell. The mechanisms used to select particular parts of the DNA or genes and template strands for the transcription operate largely at the levels of initiation and termination of transcription. Through the actions of proteins that contact DNA in a highly site-specific manner. The RNA polymerase holoenzyme makes the single-strand RNA using DNA as template. Now, in the absence of the base thymine in RNA, nucleotides with the uracil base are base paired with adenine. Like DNA polymerase, RNA polymerases catalyze polymerization of nucleotides only in the 5' to 3' direction. Unlike DNA polymerases, do not require a primer to initiate synthesis. The transcription process occurs in three distinct phases. First, initiation, elongation, and lastly, termination. Initiation and termination signals in the DNA sequence punctuate the genetic message by directing RNA polymerase to specific genes and by specifying where transcription will start, where it will stop, and which strand will be transcribed. The transcription start site or TSS is determined by the recognition of the promoter complex on the dsDNA. These promoter regions have highly conserved nucleotide sequences across species and help the RNA polymerase in recognizing the transcription start site or TSS. The region is located at the left side or upstream of the transcription start site. Once the enzyme recognizes the promoter, the large protein will lodge itself in the dsDNA, causing denaturation of the DNA. Only the region where the enzyme is found will remain denatured. When the enzyme advances along the DNA template, the DNA segment previously bound to the enzyme will hybridize and regain its double-stranded condition. So, transcription, may tatlong process pa rin. Same with replication, initiation, elongation, and termination. So class, itong illustration na to shows the process of transcription. First, initiation. Ano nangyari during initiation? Itong green na to, that is your RNA polymerase. It binds to promoter, tightly melting and opening DNA sequence that will be transcribed. Note class, nag-denature tong part or segment ng DNA. Of course, by the RNA polymerase. Next, the first phosphodiester bonds of new RNA, itong continuous na strand na to, that is the first phosphodiester bonds of the new RNA in red using a template strand where PPP meaning the triphosphate. Next step, that is the elongation. Open bubble moves with polymerase, allowing it to read and transcribe continuously, elongating the new RNA. And lastly, in termination, transcription ends when RNA polymerase reaches a termination codon. So yeah, initiation, as the RNA polymerase binds to the promoter, the open promoter complex allows the enzyme to recognize the transcription site and initiate RNA synthesis. The RNA polymerase accommodates and has a binding site for ribonucleoside triphosphates or RNTPs. The initiation process proceeds as follows. First, chain growth begins with the binding of template-specified RNTP at the initiation-specific site of RNA polymerase. The next nucleotide binds at the elongation-specific site. And lastly, the phosphodiester bond is generated by a nucleophilic attack of the electron-rich 3' OH of the first nucleotide of the alpha phosphorus of the next nucleoside triphosphate at this 5' position. The first nucleotide retains the triphosphate group intact. Next step is elongation. When a few nucleotides have been synthesized by the RNA polymerase, the sigma subunit of the enzyme dissociates and leaves the remainder of the core enzyme to continue on with the elongation process. Last step class, termination. The termination of transcription in prokaryotic systems occurs by either protein factor independent or factor dependent termination. Now, in factor independent termination, this requires two structural features that are shared by many genes. Yung GC-rich regions in the DNA, followed by an AT-rich region and a poly-A or a polyadenine region. Palindromic segments in the DNA that can result in the formation of a hairpin loop configuration in the RNA transcript that is followed by a poly-U segment. Okay, what is factor-independent termination? These features suggest the following elements of factor-independent termination. Upon reaching the GC-rich region, the enzymes slow down because of a greater number of hydrogen bonds that needs to be separated between G and C. The slow movement of enzymes allow base pairing of complementary regions in the transcript, resulting in its displacement from the DNA template. This also leads to the dissociation of the enzyme from the complex. Factor-dependent termination requires the presence of the Rho protein. The Rho protein is a multi-subunit hexameric protein that binds to the newly synthesized RNA. Once bound, it moves along the RNA until it reaches the 3' end region, unwinding it from the template and releasing the RNA. Now, eukaryotic termination of transcription. In eukaryotes, an RNA polymerase 1 is responsible for the transcription process. It continues to transcribe well past the end of the gene. After the end of the gene has been reached, it passes through one or more AATAAA sequences, which lie beyond the 3' end of the coding region. The pre-mRNA carrying this signal as AAUAAA is then cleaved by a special endonuclease that recognizes the signal and cuts at a site 11 to 30 residues to its 3' side. A tail of polyriboadenylic acid or poly-A, as much as 200 bases long, is added by a special non-template directed polymerase. The function of the poly-A tails of eukaryotic mRNAs is unknown. They are suggested to relate to message stability because the tail-less messages typically have much shorter lifetimes in the nucleus. Eukaryotic mRNAs are synthesized in the nucleus and undergo post-transcriptional processing before it is released to the cytoplasm for protein synthesis. Such processing includes capping. Ang capping is methylation of the first nucleotide at the 5' end, usually G at N7. Tailing. As earlier, polyadenylate residues are added at the 3' end. Next, splicing. Non-coding sequences known as introns are removed and only the coding regions called exons remain and are joined together. Okay, class. Translation is a process by which, or is a process whereby that information from the specific DNA sequence is expressed as a specific amino acid sequence in a polypeptide or protein. The process of translation requires a system to bring the mRNA together with the other RNA molecules such as tRNA. The system must also catalyze the polymerization of the amino acids into a polypeptide sequence. This process is done in the ribosome, which consists of 2/3 RNA and 1/3 ribosomal proteins. The message in mRNAs is the sequence of nucleotide bases and is always read in the 5' to 3' direction. The polypeptide chain is made starting with the amino end of the chain until the carboxyl end. Thus, the 5' end of the coding region of the mRNA contains the code for the amino end of the polypeptide chain. The mRNA sequence is read in single units of 3 nucleotides called a codon. The RNA molecules participating in the translation process are the major types of RNA, and they are as follows. Messenger RNAs. They carry the message from the DNA to the ribosomes to be translated into proteins. The message in mRNA is carried in groups of 3 nucleotides called codons. Each codon specifies one amino acid in a protein according to the rules of the genetic code. tRNAs or transfer RNAs provide the means of translating the genetic code. One end of the tRNA contains a 3 nucleotide sequence called the anticodon loop that is complementary to the codon of the messenger RNA. Now, the 3' end of the tRNA, which contains the CCA sequence, is covalently attached to the amino acid. Since the anticodon is complementary to the codon in the tRNA, and the tRNA carries the amino acid specified in the mRNA codon, the tRNA therefore provides the link between the nucleic acid sequence and the amino acid sequence for a polypeptide or protein in the translation process. This process that occurs in the ribosomes make a correct sequence of amino acids that correspond to the sequential arrangement of codons in the mRNA. What is rRNAs? These are ribosomal RNAs. These are component of the ribosomes where translation occurs. Now, the genetic code. The genetic code in the next slide specifies the relationship between codons and the amino acids they code. Its properties include, first, it is universal, meaning this code is almost universal, meaning that the translation process across species would be referred to this code. Non-overlapping and unpunctuated, the nucleotide sequence is always read in triplet or as one codon unit, and will not have pauses nor overlaps with other codons. Degenerate. Some amino acids are coded for by more than one codon. Unambiguous, meaning one codon will code for only one amino acid. The codons UAA, UAG, and UGA are referred to as stop codons because they do not specify any amino acid and cause the ribosome to stop translation with the help of protein release factors. The sequence AUG, which specifies the amino acid methionine, is commonly used to define a translational starting point, but AUG is also found in the middle of coding sequences. Thus, the translational starting point must have other sequences that help define where to begin. In addition, prokaryotic organisms use a modified form of methionine in the form of N-formylmethionine to start translation. Eukaryotic cells simply use methionine. Na class, itong illustration na to, this is the genetic code. Ang important dito class na amino acids and code or codons are your UAA, UAG, and UGA. These are what you call the stop codons. Dito class, pag nabasa na to ng ribosome, mag-stop na yung or mag-determinate na yung translation process. Now, kailan mag-start yung translation process? Pag nabasa ni ribosome itong code na to, AUG, which stands for methionine. Now, the entire translation process, just like transcription, includes initiation, elongation, and termination. However, unlike transcription, translation involves more molecules like protein factors for each stage. It also requires protein factors for each stage. It also requires the activation of amino acids for every transfer RNA that will bring the amino acid to the ribosome. This is an energy requiring process and involves the enzyme amino acid tRNA synthetase. Each tRNA molecule is recognized by a specific synthetase. Now, first step in translation. First, the initiation of transcription in prokaryotes involves three initiation factors: IF1, IF2, and IF3. The presence of IF1 and IF3 allows the dissociation of the 30S and 50S ribosomal subunits. IF2 is responsible for the assembly of a charged initiator tRNA, mRNA, GTP, and the free 30S subunit to form the 30S pre-initiation complex. In prokaryotic translation, the first amino acid charged to the initiator tRNA is N-formylmethionine. The enzyme transformylase adds a formyl group after the charging or binding of the amino acid to the tRNA. The tRNA charged with the formulated methionine is the only charged tRNA that can be accommodated in the pre-initiation complex. All the rest required assembled ribosomes containing both the 30S and 50S subunits. All prokaryotic systems follow this initiation process. Once the pre-initiation complex is formed, the 50S ribosomal subunit binds to the complex. The final initiation complex is now ready for the elongation phase. The complex has three sites, namely P or peptidyl sites, A or amino acyl site, and E for the exit site. The initiation codon in the mRNA and the initiator tRNA with charged formylmethionine are both aligned at the P site. The result of the formation of the initiation complex is the hydrolysis of GTP and the release of all the initiation factors. The initiation complex is now ready to proceed to the elongation phase and accept the second tRNA charged with the next amino acid specified by the second codon in the mRNA. Now, elongation phase. The 70S ribosome of the initiation complex has three binding sites to proceed to the elongation process. Figure 15 shows a single round of the process involving the formation of a new peptide bond, which is repeated until a signal for termination is encountered. The cycle goes as follows. First, the growing polypeptide chain is at the P site attached to a transferred RNA that brought the last amino acid, while the other sites A and E are empty. The codon specifying the next amino acid is aligned at the A site. Next, an elongation factor called EFTU, which carries a molecule of GTP, brings a charged tRNA to the amino acyl or A site. As soon as the next tRNA is lodged at the A site, GTP is hydrolyzed, and the EFTU GDP complex is released and used to regenerate EFTU GTP. Proofreading occurs at this step after the charged tRNA is in place. It is checked both before and after GTP hydrolysis and will be rejected if incorrect. Peptide formation comes next. The bond is formed when the growing peptide chain at the P site is transferred and attached to the amino group of the amino acid in the tRNA and the A site. This is made possible by the catalytic action of the enzyme complex called peptidyl transferase, which is an integral part of the ribosomal 50S subunit. The ghost tRNA, now uncharged tRNA at the P site through translocation, is transferred to the E site or the exit site, and at the tRNA with the growing peptide is now moved to the P site or peptidyl site. At the same time, the ribosome moves by 3 nucleotides in the 3' direction, thereby placing a new codon adjacent to now empty A site. This requires the protein factor EFG bound to GTP, where GTP is hydrolyzed in the process. As the A site is now empty, it is ready to accept another tRNA charged with amino acid specified by the next codon in the mRNA. The net result in one cycle is that the polypeptide has grown by 1 amino acid residue, and the ribosome has moved along the mRNA by a codon unit. This elongation step is repeated until a stop codon is reached, whether it be UAA, UAG, or UGA. Now, the final stage in translation, termination. The termination of translation happens when either the stop codon, UAA, or UAG, or UGA is reached. There is no tRNA corresponding to these codons, and instead of a tRNA, release factors will bind to the ribosome site. Release factors RF1, RF2, and RF3 are proteins that participate in the termination of translation. Each factor undergoes preferential binding to the ribosome depending on the stop codon aligned at the A site. RF1 binds if either UAA or UAG is present, while RF2 binds in the presence of UAA or UGA. RF3 participates more in the release process through GTP binding and consequent hydrolysis. Now, the events of termination process occur as follows. Summarize na natin to, class, ha. Depending on the stop codon aligned to the A site, release factor RF1 or RF2 binds to the A site. Next, the release of the peptide chain is facilitated by the transfer of the C-terminal residue of the polypeptide chain from the tRNA to a water molecule. The RF factors and GDP are released, and the last ghost tRNA is released. With the release of the factors, tRNA, polypeptide chain, and hydrolysis of GTP, the 70S ribosome disassembles, and it will reassociate again for another round of translation. Now, in prokaryotic translation, the polycistronic mRNA, ibig sabihin, one molecule of RNA containing more than one gene or polycistronic, may not always dissociate from the 30S subunit, but the 30S subunit just slides along the mRNA chain until it encounters the next initiation codon for another round of translation. Now, for eukaryotic cells, it is basically the same as prokaryotes. That is, messenger RNA is read by ribosomes, There are, however, significant differences, in particular, the ribosomes and the details of the translational mechanism. Ribosomes and its subunits are larger in eukaryotes. To form a functional 80S subunit, 40S ribosome, 40S and 60S subunits combine. In eukaryotic translation, initiation requires more protein factors, that is, around 11 factors are needed. Some of the initiation factors attached to the ribosomal subunits and others to mRNA. The major initiation factor is ELF2, which forms a complex with tRNA. This factor is eventually recycled via a cyclic GDP-GTP exchange called the ELF2 cycle, which requires the factor ELF2B. Eukaryotic protein synthesis is initiated as it is in prokaryotes, with a special met-tRNA reading an AUG codon, but the methionine is unformulated. The mRNA is aligned correctly on the 40S ribosomal subunit by the 5' cap, instead of the Shine-Dalgarno sequence used by prokaryotes. The ribosomal subunit then scans along the mRNA through an ATP-dependent process until the first AUG codon is found. At this point, the initiation factors are released and the 60S subunit is attached to begin translation. Elongation and termination. In contrast to prokaryotic termination, eukaryotic chain termination requires only one protein factor, your ERF, which can recognize all the three stop codons, UAA, UAG, and UGA. Otherwise, the mechanisms are very similar. Next, post-translational processing and modifications of proteins. Most eukaryotic proteins undergo post-translational modifications before they can become functional and be used for various processes in the cell. First, proteins could be folded in three-dimensional conformation of the nascent polypeptide chain through covalent disulfide bond formation and other non-covalent interactions. Next, glycosylation or addition of oligosaccharides or carbohydrate moieties. Proteins pwedeng tanggalan ng N-terminal amino acid sequences or other peptide segments by proteases, and lastly, binding with non-protein components essential to the function of the protein.
Steps of the translation process. Initiation. The small ribosomal subunit binds to the initiator tRNA carrying the initiator amino acid methionine. This complex then attaches to the cap structure at the 5' end of an mRNA and scans for the start codon AUG. The process is mediated by several initiation factors. At the start codon, the large ribosomal subunit joins the complex and all initiation factors are released. The ribosome has three sites. The A site is the entry site for new tRNA charged with amino acid or aminoacyl tRNA. The P site is occupied by peptidyl tRNA, the tRNA that carries the growing polypeptide chain. The E site is the exit site for the tRNA after it is done delivering the amino acid. The initiator tRNA is positioned in the P site. Elongation. A new tRNA carrying an amino acid enters the A site of the ribosome. On the ribosome, the anticodon of the incoming tRNA is matched against the mRNA codon positioned in the A site. During this proofreading, tRNA with incorrect anticodons are rejected and replaced by new tRNA that are again checked. When the right aminoacyl tRNA enters the A site, a peptide bond is made between the two now adjacent amino acids. As the peptide bond is formed, the tRNA in the P site releases the amino acids onto the tRNA in the A site and becomes empty. At the same time, the ribosome moves one triplet forward on the mRNA. As a result, the empty tRNA is now in the E site and the peptidyl tRNA is in the P site. The A site is now unoccupied and is ready to accept a new tRNA. The cycle is repeated for each codon on the mRNA. Termination. Termination happens when one of the three stop codons is positioned in the A site. No tRNA can fit in the A site at that point, as there are no tRNA that match that sequence. Instead, these codons are recognized by a protein, a release factor. Binding of the release factor catalyzes the cleavage of the bond between the polypeptide and the tRNA. The polypeptide is released from the ribosome. The ribosome is dissociated into subunits and is ready for a new round of translation. Initiation of translation in prokaryotes. The small ribosomal subunit is separated from the large subunit with the help of two initiation factors, IF1 and IF3. This complex then binds to a purine-rich region, the Shine-Delgarno sequence, upstream of the AUG start codon on the mRNA. The Shine-Delgarno sequence is base paired to a complementary sequence on the 16S rRNA, a component of the small subunit. This alignment ensures that the start codon is in the right position within the ribosome. Another initiation factor, IF2, brings in the initiator tRNA charged with initiator amino acid N-formylmethionine. The large ribosomal subunit joins the complex and all initiation factors are released. The ribosome has three sites. The A site is the entry site for new tRNA charged with amino acid or aminoacyl tRNA. The P site is occupied by peptidyl tRNA, the tRNA that carries the growing polypeptide chain. The E site is the exit site for the tRNA after it is done delivering the amino acid. The initiator tRNA is positioned in the P site. Elongation. A new tRNA carrying an amino acid enters the A site of the ribosome. On the ribosome, the anticodon of the incoming tRNA is matched against the mRNA codon positioned in the A site. During this proofreading, tRNA with incorrect anticodons are rejected and replaced by new tRNA that are again checked. When the right aminoacyl tRNA enters the A site, a peptide bond is made between the two now adjacent amino acids. As the peptide bond is formed, the tRNA in the P site releases the amino acids onto the tRNA in the A site and becomes empty. At the same time, the ribosome moves one triplet forward on the mRNA. As a result, the empty tRNA is now in the E site and the peptidyl tRNA is in the P site. The A site is now unoccupied and is ready to accept a new tRNA. The cycle is repeated for each codon on the mRNA. Termination. Termination happens when one of the three stop codons is positioned in the A site. No tRNA can fit in the A site at that point, as there are no tRNA that match that sequence. Instead, these codons are recognized by a protein, a release factor. Binding of the release factor catalyzes the cleavage of the bond between the polypeptide and the tRNA. The polypeptide is released from the ribosome. The ribosome is dissociated into subunits and is ready for a new round of translation.